Before start this video, please subscribe my channel and click on bell icon. Thanks.
Indiana, a team of researchers developed a technology that may detect symptoms of Parkinson's disease in urine samples. The approach allows researchers to assess if LRRK2, leucine-rich repeat kinase 2 proteins and their downstream pathways, which have been related to Parkinson's disease, are changed in Parkinson's patients' samples. The technology might potentially lead to widespread non-invasive testing for other neurological diseases, in addition to cancer. We believe this is a logical and rational approach to move forward for diagnosing Parkinson's disease, said W.A.N.D. Tao, professor of biochemistry at Purdue. Diagnosis for this type of neurodegenerative disease is difficult. Cognitive and movement testing can take a year or more to confirm the diagnosis, so molecular tests for early diagnosis and intervention can help people with Parkinson's faster, he explained. Tao and 8 co-authors from Purdue, Timora, the Michael J. Fox Foundation for Parkinson's Research, and Columbia University published their findings in the Communications Medicine Journal. It's going to be a big new area in diagnostic development, predicted co-author Anton Iliuk, Timora's president and chief technology officer, especially for neurodegenerative diseases and cancer. Parkinson's disease alone affects an estimated 1% of the over 60 population. Up to 1 million Americans live with the disease, while 90,000 new cases are diagnosed each year. The paper's co-authors include Marco Hadasuria, a doctoral student in biochemistry, Kananart Kawaranankarowan, a doctoral student in electrical and computer engineering, Xiaofeng Wu, who received his doctorate in chemistry at Purdue in 2022, Li Li, Timora Analytical Operations, Zhangqi Li, West L. Fox Foundation, which funded the work. The project began several years ago after Padmanabhan read some of Iliuk and Tao's work on the Eve trap, extracellular vesicles total recovery and purification method for urinary analysis, and proposed a collaboration. When I reviewed the data from their previous publication, Padmanabhan said, it was interesting to note the expression of an important Parkinson's disease-linked protein, LRRK2. This piqued my interest since this approach provided us with an opportunity to determine if LRRK2 proteins, or the downstream pathways they impact, are actually altered in urinary samples from Parkinson's patients who harbor a mutation in the gene. In 2017, Tao led a team that developed a blood test. In that work, Tao and his colleagues compared samples taken from breast cancer patients and a healthy control group. We identified phosphorylated proteins, which are a typical hallmark of cancers, Tao said. And within those proteins, the team found extracellular vesicles, small packages that cells use as their molecular delivery system. The finding demonstrated that a blood sample yielding phosphoproteins could serve as a potential marker for early cancer diagnosis, or for monitoring disease progression. The team was able to rapidly isolate the vesicles from urine samples, using the Eve trap method developed by Timora. We have used the method for a number of indications, primarily focusing on different cancers for biomarker discovery and validation, said Iliuk, who received his doctorate in biochemistry at Purdue in 2011. Iliuk and Tao co-founded Timora Analytical, which specializes in technology and services for detecting disease biomarkers in biofluids. This kind of analysis opens a new frontier in non-invasive diagnostics development. It's showing that biomarkers previously thought to be undetectable have become uncovered, and do a really good job of differentiating disease from non-disease state, Iliak said. It's not obvious that urine would be a source of brain-based chemicals or signatures, but it is. These EVs can penetrate the blood-brain barrier quite easily. After export from the brain into the bloodstream, they become concentrated or filtered into urine. But sampling such biomarkers from the brain via spinal tap is a highly invasive procedure. Especially for early diagnosis, that is not the preferred sampling method, Tao said. Urine samples contain proteins that could be disease markers, but many perform housekeeping functions that are unrelated to disease. Extracellular vesicles provide a way to focus on disease markers because they are released by certain types of cells, he said. Among the many ways to study the impact of LRRK2 is to track its biological pathway, which can be done by analyzing urine, blood, and cerebrospinal fluid. The EVE-TRAP method provided an easy way to track changes in urine, which is collected for many clinical studies. The LRRK2 biobanking study at Columbia University has a large bank of urine samples, available for meaningful Parkinson's disease research. Columbia University co-author Alcalay, who provided many of the samples, also helped correlate EVE-TRAP data with the clinical data. For the communication medicine study, the team studied samples from people with and without the LRRK2 gene mutation, and patients with and without the disease. Padmanabhan noted, this study also highlighted that changes in urinary proteins could serve as a proxy for changes in protein signatures that occur in brain diseases like Parkinson's disease. Thanks for watching. Please subscribe my channel for more videos.